The ABO blood group has what we consider to be subgroups. Basically, they are groups within a group. That is basically the idea of a subgroup. The original report of most ABO subgroups were made before the availability, as we discussed previously, of the monoclonal typing reagents, all right, which are currently used in routine ABO blood grouping, which is one of the most common testing procedures that are done in the blood banking laboratory. So the ABO subgroup represent the phenotype showing weaker and variable serological reactions with the commonly used anti-A and anti-B and anti-AB reagents that are currently being used in our laboratories today. But the subgroup that I want us to begin our Cavernous investigation concerning is the A subgroup. Yes, remember we spoke about A1, we spoke about A2, and we made an allusion to A3. But guess what? There are more subgroups of A. So it was in 1911 when von Dungerms described two different A antigen based on reaction between A red blood cell and anti-A and anti-A1 lectin. So the A1 is the one that is predominantly observed in our population, and that is why we use the A1 lectin to take from the A people to get those who are A1, and we pool those together, and then we have our antisera. But this A1 lectin is derived actually from a plant base, which we'll get into in a little while. All right. So group A red blood cell that react with the anti-A and the anti-A1 are classified as A1, right? Because they react with A, anti-A, and usually if you react with A, it means that you have the antigens on your surface of your red blood cell. And if you have the A and then we react to you with the A1 lectin and you give a positive result, which means that there is some agglutination taking place, it really indicates to us that you are positive for A1, which means that you have the A1 version of the A. All right, so that is just a simplified version of the whole process of approach, right? So if a person doesn't have the A1, what do they have? So whereas those who react with anti-A and not with anti-A1, these people are usually classified as A2. Yes, A2. So we know A1, now we have been exposed to A2. So the red blood cell from A1 and A2 individuals reacts basically equally strong. So yes, there is a reaction that is observable, and we can use this to determine the blood type of the individual. So they react with the current reagent monoclonal anti-A in the forward grouping. So in the forward grouping, because they have the A antigens on the cells, it can be identified with the use of the monoclonal anti-A, which is in the vial, and it comes in a blue color. All right. So the A subgroups are more common than B subgroups. So yes, we are alluding to the fact that A subgroup exists and we also have B subgroups that exist. So the A subgroups are more common, and we have seen both A and B in practice. The weaker serological reaction of the ABO subgroup is attributed to the decreased number of A and B antigen sites on the individual's red blood cell. So depending on how many antigen binding sites you have on your red blood cell, that would give an idea as to the reactivity and the expression that will be observed, right? So the classification of the A into A1 and A2 phenotype accounts for 99% of all A individuals. So if you know the A1 and you know the A2, guess what? You know 99% of what you need to know about the A people. All right. So the cells of approximately 80% of A or AB individuals are A1, or if they are AB, it could be A1B. All right, because with AB you have A and B being expressed. All right, or if it is the A person, it is just A1. Now, the remaining 20% are A2, or for the AB person, it would be A2B. All right, so understanding this based on the results that we get from our typing and the use of the A1 lectin, we can determine if a person is A1 or A2. If the A1 and the A2 are not differentiated, then we'll just put A or AB, right? But if we know, we use the A1 lectin and then we get a positive reaction and the person is A, we know that this person is A1. If we use the A1 lectin and we get a positive reaction with the AB person, we know that this person is A1B. All right? And if it is not positive when we use the A1 lectin, then we know that this person is A2. And if it's an AB person, then the person is going to be A2B. All right? And these tend to react a little bit weaker. So the difference between the A1 and the A2 are both quantitative and qualitative. So quantity, when we talk about quantitative, and quality in terms of how they interact. All right, so just bear that in mind. So if we look at the quantitative and qualitative differences of A1 and A2, we can look at the number of antigen sites, the amount of transferase enzymes, the amount of branching that is taking place. For qualitative, we can talk about the difference in the precursor oligosaccharide chain. We can talk about the subtle difference in the transferase enzymes. We can also talk about the formation of the anti-A1 in a percentage of some subgroups. All right, so these are just some things that we should bear in mind as we continue our investigation on the A1 and A2. So we are talking and we are working our way around to a complete understanding. But in order for us to grasp everything, we need to remember that the production of both types of antigen is a result of the inheritance of the genes on the ABO. So A1 individuals or A2 individuals, the gene is inherited on the ABO locus, right? So inheritance of the A1 gene elicit the production of a high concentration of enzyme alpha 3 N acetylgalactosamyl transferase. All right, remember we said that the A's are harder to pronounce, so yes, this is the A again, which converts almost all the H precursor to A1 substances. All right, on the red blood cell we're talking about. So they are very potent in terms of their expression. They created about 810,000 and 1,170,000 antigen sites in adults with the A1 red blood cell, whereas the inheritance of the A2 gene result in a lower production, in which we have what we consider to be maybe 240,000 to approximately 290,000 antigen sites on the adult A2 red blood cell. So this is something that is very interesting, and it gives us an idea as to the epitope and to the antigen binding sites that are available. And based on the availability, we can determine what is the expected reaction that we can perceive. All right? So, the A2 allele is characterized by a single base substitution at the nucleotide 467 and a single base deletion at the nucleotide 1060, and we normally refer to it as 1060 DEL deletion of this nucleotide. All right, so this is what we normally refer to it as. And remember, most of this reaction takes place in 6 and 7, but in this situation, it is taking place in exon number 7. All right, so let's just do a quick recap again for the antigen binding sites. So for the A1, which we made mention of, it has 810,000 to 1,170,000, right? Then the A2 has less antigen binding sites. You have 240,000 to about 290,000, right? And this is in adults, in adult adult humans. So we have a nice understanding of what is taking place. And let us not forget the sugar. So it's alpha 3 N acetyl ACETYL galactosamine nil transferase, TRANSFERASE. All right, beautiful. So now we have that documented, and we have a visual representation imprinted in our mind. All right, so these substitution alters the activity of the site specificity, and when that happens, there is a challenge in the coding region, and the A glycosyltransferase is diminished in some instance. The immunodominant sugar on both the A1 and the A2 red blood cell is the N-acetyl D-galactosamine. All right. So the dominant sugar, let me just DOMINANT sugar for the A1 and the A2 is N-acetyl, C-E-T-Y-L-E, D, don't forget the D, the N, the D, and gal, lactose amine. All right, so now we have that. So the qualitative differences that exist, because there are quantitative and there are qualitative difference. So since 1 to 8% of the A2 inherited individual produce anti-A1 in their serum, and 22 to about 35 of the A2B individuals produce anti-A1. So even though these individuals are A2 majority, they still have some level of A1 that exists. So this gives us a little idea as to the technicalities that are about to unfold. So this antibody can cause discrepancies between the forward and the reverse blood grouping. So when we're doing the blood typing, we may get one reaction in the forward and we say, yes, this person looks like an A, and then when we check out the reverse is saying that this person is O, because there's no reaction, or there is a problem in the interpretation. In the forward, we may get A, and in the reverse, we may get that, hey, this person may be B, and then that there is problematic. So there are many different kind of discrepancies that can occur. All right, but we have to be mindful to know what to expect, and when we know what to expect, then we can move into performing the relevant procedures in order to get that which is expected, and to determine why we are not getting what is expected. So this system of the A1 and the A2 subgroup is very intense, but it is something that every student should know and understand. So the anti-A1 is usually naturally occurring, which means that, as we stated before, when we are born, we already have our antigen, the binding site. They just mature a little bit more when we come to maturity, 2 to 4 years old. All right. So what we need to remember is that these antis, they are IgM. So the anti-A1 is an IgM, cold reacting antibody, and is unlikely to cause a transfusion reaction, because the body is usually at about 37 degrees when blood is being transfused. So if you have a cold reacting and your body is already at 37 degree, then the likelihood of a transfusion reaction from the activation of that antibody is negligible. All right. So let us bear in mind that the body is at 37 degrees. We normally transfuse blood at 37 degrees. So the blood is warm and it is transfused. So the antibodies that will be considered to be clinically significant in terms of can mount or evoke a transfusion reaction would be those that react at 37 degrees. All right? So just bear that in mind so that you can navigate your notes and have a better understanding and appreciation for what is happening. So the antigen present on the red blood cell of the A1 and the A2 individual can be depicted in two ways. And this depiction, most often A1s are illustrated as having both A and A1 antigen on the cell surface, in contrast to the A2 cells, which only contains the A1, the A rather. All right, so the A2 cell only contains the A. Alternatively, the A1 red blood cell can also be conceptualized as having only A1 antigen sites and the A2 also only having A antigen sites. But in routine forward grouping, reagent anti-A strongly agglutinates both A1 and the A2 phenotype. Differentiation of the A1 and the A2 phenotype is determined serologically using the A1 lectin. And this A1 lectin, this is very important for us to know. The A1 lectin is made from a seed of a plant. It is called the Dolichos biflorus. All right, let me write that down because I have had many students who have seen this question come on the ASCP examination. All right, they ask about the A1 lectin and its derivative, where it is derived, and it is from a seed. All right, so seed, that's a seed, SEED. All right, that's where it comes from, and the name of the plant is Dolichos, Dolichos biflorus. So it's BIFLORUS. All right, and this is all italicized. All right, so this is a plant. So let us just bear that in mind. So the lectins are seed extracts that agglutinate human cells with some degree of specificity. The A1 lectin agglutinates A1 or A1B cells, but does not agglutinate A2 or A2B. So one is for the A group and one is for those who are AB. All right, so we have that all clear. Due to the A2 glycosyl transferase being less efficient at adding the immunodominant sugar to the H antigen precursor, the A2 red blood cell shows an increased reactivity with the anti-H lectin. All right, and this is very important for us to know. So we have what we call the anti-H lectin. So anti-H lectin, and this has a very special name as well. I want you to remember this name. It's called ULEX, ULEX, and Euro PAEUS, ULEX europaeus. All right, that's how we normally pronounce it, but if you have a different pronunciation, please use it. But the idea is that you know how to identify this this name and its association. All right, so ULEX europaeus. All right, and this here is the anti-H lectin. So H antigen is found in a greater concentration on the RBC of group O individuals, all right, because they did not differentiate to produce A or B, so they have just the H. And in the ABO blood grouping, we use anti-A, anti-B, anti-AB for the forward grouping. We don't use anti-H unless we're doing some investigation. So if we have an O individual, we are expecting that there will be a predominance of H antigen that is found on that individual. So the H antigen may not be detectable in the A1 individual because of the presence of the A1 gene, which almost all of the H antigen is converted to A1 antigen. And this conversion takes place by a sugar reaction. All right. So the H antigen is converted to A1 antigen by placing the large N-acetyl D-galactosamine sugar on the H substance. All right. So due to the presence of so many A1 antigen, the H antigen on the A1 and the A1B red blood cell may be hidden and therefore may not be available to react with anti-H antisera. All right, because if there's none present, or if all of them have been used up, or if the titer is too low based on the volume that is available, then you can expect a reaction to be almost non-existent. All right, so just bear that in mind and bear the concepts of how we come to the understanding in mind as well. So in the presence of the A2 gene, only some of the H antigen is converted to A antigen, and the remaining H antigen is detectable on the cell. So if you use the anti-H with the A2, you will be able to find or get a stronger reaction with the anti-H than you would if you did it with the A1 individuals. So weak subgroups of A antigens will often have an inverse relationship between the amount of H antigen on the red blood cell and the amount of A antigen formed. The H antigen on the A1 and the A1B individuals is so well hidden by the N-acetyl D-galactosamine that the anti-H is occasionally found in the serum. This anti-H, guess what, is naturally occurring. So we are thinking about IgM cold agglutinin that reacts best at room temperature. As can be expected, this antibody is formed in response to a natural substance and reacts most strongly with cells of group O individual. So just bear that in mind as you navigate the concepts of the A and A subgroups. All right, so let us create a mental picture, a mental representation of the information that we have just received. All right, so we have established that we have two variants in terms of the phenotype of A. So A subgroup, let me just write it, subgroup, Group. All right, we have the blood group, and then we have the anti-A reagent, because we're going to react anti-A reagent with it, anti-A reagent, which is yes, and then we're going to use anti-A1 lectin, anti-A1 lectin. All right, so a A1 person is going to have a reaction with the A. Let me all right. So the A1 person is going to have a reaction with the A and also a reaction with the A1 lectin. An A2 individual is going to have a reaction with the A, but they will not have a reaction with the A1 lectin, because the A1 lectin is specific for A group with the A1. All right, so that is all you need to have an appreciation for at this time. All right, so let us try to do a reaction to determine the A substances. So if the blood group is A1, what antigen? Let's do, this is a blood group, and then we're going to do the antigens present, and then its reaction with the antisera, which is the anti-A, and anti-A1 lectin. A1. So this is the A1 lectin. Bam, bam, bam. Alright, so this person is A1. So if they're A1, we expect them to have A1 and we expect them to have A. Right? They're supposed to have a reaction with the A as is, and they're supposed to have a reaction with the A1 lectin. If they're A2, we're expecting them to have A, but they won't have the A1, and we're expecting them to react with anti-A, and we are not expecting any reaction with the A1 lectin. All right, so that is the simple essence of the reaction profile that is expected for an individual with A1 or A2. All right, beautiful. So now let us move in a little further to finish our investigation. Let us try to do a forward and a reverse blood grouping, or yes, I think we have time. We could do a little forward and reverse blood grouping. So the person is A1. This is a phenotype. Let's do the phenotype. The anti-A, so that's anti-A, and this is anti-B, and this is anti-AB, right, and anti-A1. So we're just going to do this section first, and then we're going to move down and add. Let's do the A cells and the B cells. All right, so if a person is A1, we're going to expect a reaction in the A, no reaction in the B. We're going to expect a reaction in the B, AB rather, and we're going to expect a reaction in the A1, right? So if you have A antigen, you're not going to have A antibodies, you're going to have B antibodies. So no reaction and 4+ reaction, all right? So that is just basically what we're expecting. So the person is A2. We are going to also expect a 4+ reaction. We're going to expect a reaction in the A. We're not going to expect anything in the B. In the AB, we're going to expect something. Anti-A1 lectin, there's not going to be any. A cells, there's not going to be a reaction. Then we're expecting there to be a reaction in the B, maybe 3+, 4+. It all depends on the individual, all right? So that there is for us to have a appreciation of. So we recall that there are different antigen binding sites. So the A1 would have about 810,000. This is antigen binding site, and 1,170,000 approximately, all right? And the A2 would have about 240,000 to 290,000. Right, so this is just for us to wrap our minds around the information. So A1 and A2, there is substance that is present in the saliva, and let me just put it right here, substance present in saliva. In saliva, and that would be the A and the H. So that is in A1, and the same thing in A2. So just keep that in mind, all right? So we were discussing previously that there is a difference in the reactivity of the anti-H antiseria or the anti-H lectin with the ABO, all right? So I want you to bear this in mind. The O individual has the most amount of H, all right? So O has the greatest amount of H, greatest amount of H. And the A1B individuals has the least amount of H. So this H, right? So this is the most, and this is the least. So A2 individuals have less than the O, right? But the B individuals have less than A2. Guess what? The A2B individuals have less than the B. And the A1 individual have less than the A2B. And the A1B has less than the A1 in terms of the presence of H. All right? So just bear this in mind so as you can know what to expect with your different investigations that you may undergo.